Hello everyone. Today we are going to see the acid-fast staining. Acid-fast bacteria, otherwise said to be acid-fast bacilli or AFB, or a group of bacteria sharing the characteristic of acid fastness. Acid fastness is a physical property that gives the bacterium the ability to resist decolorization. Means due to the presence of mycolic acid, they resist to decolorize once they take up the stain. By the acids, it can be a sulfuric acid or a hydrochloric acid. There is a specific percentage during staining procedure. This is acid fast staining is used for specifically TB microscopy. So the smear is said to be acid fast smear. The microscopes that can be used are compound microscope, or if the staining procedure adapted is a fluorochrome staining, then a fluorescent microscope is necessary. The TB diagnosis using this characteristic is referred to as TB microscopy, of course. Even though the use of highly advanced molecular diagnostic test has come into play, the value of this staining technique cannot be overstated, especially for low and middle income countries, because it is considered to be standard, highly sensitive and cost effective. The principle of this staining, it is a differential staining. You would have known that gram staining is also one of the differential staining. The other example is acid fast staining. That's what we are seeing now. The ordinary aniline dye solutions do not readily penetrate the substance of the tubercle bacillus because it possesses mycolic acid, which is a waxy substance. Means the aniline dyes, in the sense the crystal violet, methyl violet and all, will not be taken up by this organism and therefore unsuitable for staining it. But we can make the organism to get stained by using a strong solutions like phenol or heat, which acts as a lipid solvents, so that enables the stain to penetrate the organism and it takes up the stain. Once it is stained, that cannot be easily decolorized. So this makes a special property for us to make it to differentiate from other non-acid fast organisms. So once stained, the tubercle bacillus will withstand the action of powerful decolorizing agents for a time period and retains the stain. The red color will be usually used when everything else in the smear will be decolorized and take up the counter stain like methylene blue or malachite grain. So in the interpretation, we will be seeing the acid fast organism to be in red color and the background, which possesses polymorphonuclear neutrophils or the mucin layers or other bacteria which are non-acid fast, to be blue in color. So I think you would have understood now why the bacilli alone is red. We allow the organism to take up the stain by providing heat or by giving phenol so that it takes up the stain. By using the decolorizers like 20% sulfuric acid or 3% of hydrochloric acid, we are decolorizing the smear, but the acid fast organism alone retain the smear at that percentage for a particular time period. So when we use a counter stain, the background takes up the stain, the counter stain blue color, whereas the acid fast bacilli still remains with the pink color. Here I have given you a peculiar characteristic, the types and the procedure of how to perform the acid fast staining. Basically, we will be having primary stain that will be proceeded with the decolorization technique and the counter stain. The primary stain will be usually carbonyl fission. This can be a carbonyl fission, concentrated carbonyl fission, or Quignon's carbonyl fission, or Oramin phenol stain in a case of fluorochrome staining where we need a fluorescent microscope. This is for Mycobacterium tuberculosis. If we use the carbonyl fission alone, then we need the heat. If you use the Quignon's carbonyl fission, there is no need of heat because phenol acts as a lipid solvent there. And when we come to the decolorizer for Mycobacterium tuberculosis, usually it is 20% sulfuric acid. If you are going to differentiate Mycobacterium tuberculosis from other Mycobacterium species, then we can use the acid and also alcohol. Here is 95% ethanol. Mycobacterium tuberculosis is both acid fast and also alcohol fast, whereas other Mycobacterium species are only acid fast. This also helps us to differentiate the species of Mycobacterium. The counter stain is of malachite green or the low flush methylene blue, as I have told you before. When you come to the Mycobacterium leprae, the primary stain remains the same as like our Mycobacterium tuberculosis. It can be a Ziehl-Neelsen method or a Quignon method. Ziehl-Neelsen means with the carbonyl fission. Quignon's method means with the Quignon's carbonyl fission. The heating is required as like our Mycobacterium tuberculosis, where Ziehl-Neelsen method requires heat, Quignon's method does not require heat. The decolorizer is different when compared to the Mycobacterium tuberculosis. Here 5% sulfuric acid alone can be used, whereas in Mycobacterium tuberculosis is 20% sulfuric acid or 3% hydrochloric acid. Your counter stain also the same like our Mycobacterium tuberculosis. It can be a malachite green or low flush methylene blue. Coming to Nocardia and other acid fast parasites, here the primary stain again is the same. It can be a Ziehl-Neelsen method with carbonyl fission alone or Quignon's method with Quignon's carbonyl fission. Here again heat can be required for Ziehl-Neelsen method, whereas for Quignon's method no heat is required. But the decolorizer is only 1% here, that is sulfuric acid, no HCl, no 20% or no 5%. Here the counter staining is the same, like malachite green can be used or low flush methylene blue can be used. There is another one method where we can adapt decolorization plus counter staining in one step. That is said to be Gabbett's methylene blue, which will have the combination of decolorizer and the counter stain together, so that our steps become very easy to go with. So I have given the formulations for primary stain here. You can take note of that, where the Ziehl-Neelsen carbonyl fission staining combination, the composition is given, Quignon's carbonyl fission composition is given, Oramin composition is also given. There is a formulation for decolorizer. Specifically I have given for 20% sulfuric acid, 5% sulfuric acid, and you can make the same for 1% also, like 10 ml to 990 ml of distilled water, 10 ml of concentrated sulfuric acid and 990 ml of distilled water. Here 95% alcohol is also given. Industrial methylated spirit can be replacing to our 95% ethanol. Acid alcohol decolorizer combination is also given here. This is used for differentiating Mycobacterium tuberculosis from other Mycobacterium species. Here counter staining formulation is also given, low flush methylene blue and malachite green. Interpretation is very important in acid fast staining. Even the smear preparation is also important. There is a special process how to prepare a smear. I'll put a separate post for how to make a smear for acid fast staining. Now coming to the interpretation part, here the acid fast bacilli appears pink color, the background will be blue, I have shown in the picture. So we have a National Tuberculosis Elimination Program guidelines, NTEP guidelines for interpreting the same smear. Here there is a different procedure how to adapt for our compound microscopy, and also by fluorescent microscopy the interpretation varies. Sensitivity is more with the fluorescent staining because we are enabling maximum number of fields with 40x objective lens, whereas in the compound microscopy the interpretation should be done only with the 100x oil immersion. So sensitivity is more with the fluorescent staining. Coming to the Mycobacterium leprae, we have bacteriological index and the morphological index for reporting the same for Mycobacterium leprae, which causes our Hansen's disease or leprosy. Both the tuberculosis interpretation chart, the guidelines, and also the Mycobacterium leprae guidelines chart is utmost important to check the prognosis of the patient, which is directly related to our treatment, the drug of choice. Thank you very much. If you like the video, share, subscribe and click the bell icon. Thank you.